Hi, welcome everyone. We'll get started in just a moment. Hi, welcome everyone to today's webinar, presenting the winners of the 2026 Ocean Innovator Award, sponsored by Sea and Shore Solutions. My name is Sarah Carr, and I'm Chief Knowledge Broker for Octo, Open Communications for the Ocean. Octo is co-hosting today's webinar with Sea and Shore Solutions. Before I turn you over to our organizers from Sea and Shore Solutions, we have Adrian Laufer, Deanna Caracciolo, and Katie Bernalvin. I wanted to let you know how to ask questions during the webinar. We'll have presentations from today's award winners, and then we will have dedicated time for Q&A. But we encourage you to send in your questions at any point during the webinar, and we can hold them till the end. So there's two ways you can ask questions. You can send them in through the question and answer panel at the bottom of in the Zoom user interface, or you can send them in through the chat. With the chat, you have the option of making chats visible to all the attendees, and you're welcome to use this option, especially if you're interested in something input from other attendees, but we just ask that anything you put in the chat be on topic. So that's all from me, and I'll turn you over to Adrian next, and welcome everyone.
Yeah, thank you so much, Sarah, and welcome everyone. Thank you for joining us today. We are so excited to be here today with Octo to feature our amazing winners of the 2026 Ocean Innovator Award. As Sarah said, my name is Adrian Laufer. I'm the CEO and co-founder of Sea and Shore Solutions, or SNS. We're honored to be here today, and I will start us off just by writing a little bit of background about SNS, who we are, some housekeeping, and then we'll quickly hand it over to our amazing winners so they can share about their work. So some housekeeping first. All attendees will be muted throughout the webinar with your videos turned off. When we get to the Q&A, there might be opportunities for folks to come off mute and show their camera to ask questions live. Closed captioning is available in the Zoom platform by clicking on the CC button in your little info panel. As Sarah mentioned, there will be a Q&A at the end. You can use either the chat or the Q&A function. And if you have any technical issues throughout this webinar, Sarah is going to be available to help you. So you can either chat her within the Zoom interface directly or email her at sarah@octogroup.org. So a little bit about SNS. We are a women-owned environmental consulting firm focused on creating and implementing innovative solutions for coastal, marine, and estuarine spaces, including species, I guess. Our team provides high-quality, innovative solutions that bridge the gap between best available science and application. And that kind of falls into four main buckets: management, in policy, through science communication, and then just generally through organizational support for all the different firms that are working in this space. As a company, our work is informed by 5 core values: collaboration, strategy, science, innovation, and outcomes. So each year we run the Ocean Innovator Award as a way to dig into our innovation value. This award serves to increase the visibility of high quality innovative solutions and to support early career professionals. We host this competition yearly, seeking creative applications from early career professionals in coastal, ocean, and estuarine fields that showcase innovative science or projects that contribute to sailing and ocean topics. Projects can be in any stage of the process to apply, from ideation to being in the middle of the process all the way through completion. Applications are then reviewed by a panel of experts with diverse specialties within the field, and the winners are awarded up to $2,000 and are showcased in this webinar. Since this program was started in 2024, we've received dozens of applications from across the globe, hundreds of applications from across the globe, and have awarded several winners. We're excited to see this program spread to new regions each year, such as Mozambique, India, the Netherlands, Iran, and even as far as New Zealand. The ocean is facing global challenges that require global solutions, so we hope to continue to get more global applications throughout time. So the next application period will open in February 2027. So Deanna is now putting a link in the chat for anyone that would like to fill out this short form. We'll send you updates when things are posted about this next cycle if you are interested in potentially applying. Great. And with that, I will introduce our amazing 2026 Ocean Innovators. We have Andrea de Moura Milanelli, third place, Yudmila Chunguane, second place, and Wave Moretto in first place. And now I will pass it over to Katie to introduce each of our winners.
Thank you so much, Adrian, and welcome everybody. My name is Katie Bear-Nalvin. I'm a project coordinator here at Sea and Shore Solutions, and it's my honor to introduce our first speaker, Andrea Milanelli, as she gets her slides pulled up. Andrea Milanelli is a marine biologist and early career researcher whose academic and professional trajectory has been shaped by a strong interest in using sound to better monitor and understand marine life to support conservation. Her research interests include marine bioacoustics, soundscape ecology, and conservation. She has a wide range of experiences, including trials on Icelandic fishing vessels, dolphin observation, photo identification, acoustic monitoring, and even marine animal rehabilitation. Andrea completed her MSc in biological oceanography at the Federal University of Rio Grande in Brazil, and now she is currently a research assistant and PhD candidate at the Thünen Institute in Rostock, Germany, where she works on cetacean bycatch mitigation within the Fishing Gear Technology Working Group. Her current research focuses on the development of modified gill nets with the integrated acoustic reflectors to reduce odontocetes bycatch, combining acoustic simulations and echo sounder measurements to optimize reflector design for effectiveness, durability, and large-scale application. It's my pleasure to hand it on over to you, Andrea.
Hi, thank you for the introduction. So today I will talk about the PearlNet as a potential bycatch mitigation tool for toothed whales in gillnet fisheries. So our focus species is the harbor porpoises, these small cetaceans that because of their preference for coastal habitats, they are very vulnerable for bycatch in gillnet fisheries. And one of the reasons could be because they cannot see the nets using the echolocation. So they can only see the top and the bottom lines. So they think that they can pass through it. So what we should do in this context to make the net more visible for them, so they can identify it as a barrier and avoid it on time. So we do that attaching acoustic reflectors to the netting, and the definition of these optimal acoustic reflectors needs to fulfill some criteria. So we cannot harm the fishery, but we need to keep the acoustic properties. So we need an object that has a strong echo, needs to be omnidirectional, so it doesn't matter where the animal is coming from, needs to be small and neutrally buoyant, so the net won't change the underwater behavior. So this is our final product. So we have these small spheres that are made by acrylic glass that we call pearls. And this is the PearlNet. This is how they look like. They are only 8 millimeters diameter. They are very small. So we know that this works from the physical point of view, but we also want to understand how these animals perceive the net. So we went out in our research vessel and we set up the PearlNet and standard nets, and we were using an echo sounder with the harbor porpoise frequency to see how is the impression that we have. And we can see clearly the difference between the two gears. So while in the standard net, we can see the float line and the lead line, and we can see anything in the middle, while in the PearlNet, we can see reflections all over it. This indicates that the animal would be able to detect the PearlNet as a barrier. And the most fascinating thing is that we are using 8 millimeters because this is the pearls. They have the size of the pearl is strictly related to the frequency, the click frequency of the animals. So as soon as we have a match between the diameter and the frequency, the pearl starts to resonate like a guitar string. So we can use this for different species by just adjusting the size of the spheres. So it's a wide tool that we could use in different places around the world. But yeah, we know the theory, and what about if the PearlNet is working? Before answering this, we need to make sure that the PearlNet is not affecting the fishery. So this means that the fishers cannot spend more time handling the net, and also that the PearlNet is not catching less fish than the standard net. So we tested this in Iceland, Denmark, and Germany, and our results were very positive. So the PearlNet is not affecting the fishery. So now we can move on. What about the bycatch reduction? So we went to Iceland in a commercial fishing vessel, and we were replacing part of their nets with the PearlNet. So the strings, they had the same configuration, except that the PearlNet has had the pearls attached, and they were a bit smaller. Our results, after 7 days, we had 20 bycaught animals in the standard net and 1 porpoise in the PearlNet. But we need to be careful because we had way much more effort, fishing effort with the standard net. So when we standardize it by net length, we have for every porpoise caught in the PearlNet, we have 4.4 porpoises in the standard net. This means a reduction of 77.4%, which is really good. But for the statistical point of view, the bycatch is a rare event. So our statistical tests, we don't have the significance that we were aiming for, but I would say that we have a strong tendency, and these results, they are very similar with the one found in Turkey. So they also had a reduction there, but again, they didn't have the statistical power. So I would say that we are halfway through it. But as every new technology, the PearlNet also has its limitation. So our main limitation now is regarding the attachment of the pearls in the net, because our current design is a sphere with a slit, so we are gluing them one by one. And this is a very time-consuming production, which makes the net more expensive. And also the glue, it's a very challenging process, because if you add more glue than you should, and this glue is drying like over the pearl, this means that you can have more entangled than you should be. So this is not good for the fishery. So we need to change. We need a new design, and we are starting our process again. So we need a full sphere, if possible, that enable a smooth assembly. We need to have a smoother outer skin, so no elevations or depressions, and ideally asymmetric shape. So after many tests, we have the new pearl that we call the grip pearl. So we have 2 half spheres, they are exactly the same. So the attachment is way much faster. You just need to click them. It's very fast and very efficient. And as we are changing the design, we are also thinking about new materials, because one of the questions that is always coming to us is about the marine litter. So we were thinking, what about the biodegradable plastics? But as the name says, when the plastic starts to decompose, the acoustic properties will change, so we can't control that. So what about making the recycling process easier? Because the majority of the nets are made by nylon. So if we are doing pearls with the same material of the netting, so the recycling process would be easier. And because now we have the grip pearls, and the acrylic glass is a very stiff material, so when you attach them together, the chance of breaking is very high. We are also thinking of making new pearls with polycarbonate. The polycarbonate is it's more malleable material, so it would be better for this new design. So we went again in our research vessel, and we are doing acoustic measurements of the grip pearls in acrylic and polycarbonate, and also nylon pearls. And we wanted to compare these results with our actual pearl, and these results are still ongoing. So stay tuned for the news. And now we need you. So we need to test the PearlNet in new fisheries. So if you know a place that you have conflicts between fishers regarding bycatch of toothed whales, please let me know. I would be very happy to collaborate. Also, we really want to understand why these animals are getting caught. So if you work with acoustics or behavior, please let me know. We can think about a potential collaboration with this. So I would like to thank you, all the partners, all the co-authors. It's a very international project with a lot of collaboration. Also the founders, and of course the Sea Source Solutions for the opportunity, and Octor to organizing the webinar. And thank you so much.
Thank you so much, Andrea. That's a lovely presentation. It's so great to hear more about PearlNet and all your amazing work. As a note, please add any questions that you may have for Andrea into the chat or the Q&A, and we're going to save those for the end of the webinar. So now it is my pleasure to introduce our next winner as they get their slides up. Yudmila Chunguane is a marine biologist from Mozambique with a background in marine, aquatic, and coastal biology. She is currently finishing her MSc in conservation biology and ecology. Yudmila has been involved in a wide range of conservation projects across Mozambique, working with different ecosystems and species, from coral reefs to mangroves to marine megafauna. Yudmila's work is driven by a strong interest in understanding marine ecosystems and finding practical community-based solutions for their conservation. She is particularly interested in coral ecosystems and their connection to food security, particularly in Jangamo, where her team at Love the Oceans are based. Through her academic and professional experience, she has developed a multidisciplinary approach that combines science, conservation, and community engagement. Yudmila, in addition to now being an ocean innovator, is a Darwin 200 leader, a Conservation Nation grantee, a Conservation Leadership Program grantee, and a certified PADI rescue diver. She is the project leader and coral biologist at Love the Oceans, where she forms part of a core team dedicated to protecting marine life in Jangamo Bay. Love the Oceans work closely aligns with Yudmila's values, following a community-led holistic approach to benefit both local populations and the ocean on which they depend. Love the Oceans work involves coral reef, fisheries, and megafauna monitoring, as well as community outreach and initiatives focused on sustainable livelihoods, ocean literacy, and gender equity. It's now my pleasure to hand it on over to you, Yudmila.
Thank you, Kate. Hello, everyone, and thank you for the wonderful presentation, introduction, sorry. Hey, everyone again. My name is Yudmila Chunguane. I'm a marine biologist from Mozambique. I work for Love the Ocean, as Kate said, and is a grassroots conservation charity based in Jangamo, Mozambique. Love the Ocean has mission to create a holistic conservation strategy that can be replicable, scalable along the coastline of Mozambique and beyond. And today I'm going to talk about one of our projects, which is the project that I am the lead, that's related to the low-cost coral reef restoration, which is the project BEAM, that stands for Biodiversity, Enhancement, Algal Management. This project is located in Africa, Mozambique, in a province called Inhambane, in a specific community called Ginjata. And this project is located as well within the area that Love the Ocean is working towards establish as a local managed marine protected area. And it's important to refer that as Mozambique has a long coastline, a lot of people depend on the ocean for resource, food, and livelihood. And not being different from that, the Ginjata community, there is a community of fishermen. So the fish is their only source of protein. And fish are linked to corals. because corals are this essential ecosystem that support the marine biodiversity. They serve as a nursery ground for a lot of marine species. They protect the coastline. And apart from the other ecological service that they provide, they also sustain the livelihood of the coastal community. And the corals in Ginjata or in Jangamo, they are facing a huge threat, which is the algae overgrowth that tends to suffocate the corals. And this is happening because of the overfishing of the population of herbivorous fish, like parrotfish and the rabbit fish. And that in these species, they feed on algae and they control its growth. And without these species, the algae grows rapidly, taking over the coral, and also, as well, disrupting the coral recruitment. One of the Love the Ocean project looks at as well the cryptic species, not cryptic species, and the benthic cover. And these long monitoring data have shown that algae have more than doubled between 2018 and 2024. And this threatens the coral health. It creates a huge impact on fisheries and as well in the food security of the Jangamo local community. Okay, to address that, Love the Ocean brings an innovative and low-cost restoration technique known as seaweeding. With this technique, we will remove manually the algae that is choking the coral and from selected plots. You can think of it like weeding your garden, but underwater. It's a low-cost approach because we are going to use our hands, we are going to use scrapers and brushes. And this project is happening in collaboration with scientists from James Cook University in Australia, which this technique has shown promising result, has a huge success, and they publish a lot of articles related to that. So this make the first trial technique outside Australia, making Mozambique a key testing ground for a scalable and accessible restoration technique, especially for countries that have limited resources in terms of financially, in terms of access to the laboratory, and so on. In terms of methodology, we meant to tag 40 coral colonies, and in the dive site, in one of the dive sites that we mostly dive. And from this, we are going to have 20 coral colonies that will act as a treatment, where we are going to remove the algae in the immediate area that is fighting against the coral, and 20 colonies for control that we are not going to remove, and we are going to monitor it. In terms of species, we are working with the Goniastria and Poecilophora genera. And as I told, we are going to remove the algae that is in the immediate area around the coral. And we are going to take photos before and after this cleaning. And we are going to use ImageJ to be able to calculate the area. And with these photos, we are going to keep monitoring to be able to understand if the coral is growing, if there is any recruitment, and if there is any improving at all. For the first part of the project, we started a campaign and a big gift where they doubled the amount that we raised, and this money was allowed us to start preparing this the restoration plot. But unfortunately, challenge condition during dives, like surges and currents, meant that we needed more dives that we expected. Mozambique as well experienced floods in January, and in February we are hit as well by Cyclone Jazan that distracted a lot of electricity poles. So basically the funds were not sufficient to finish the project. That's why we are so happy, and I'm so happy to be able to win this award and be able to continue with this project. go. From ecological, sorry, my computer is going faster than it meant to be. Apart from ecological restoration, Project BEAM also focused strongly in local capacity building. We are training local community as a divers, as a reef monitors, as swimming instructors. We are delivering as well free swimming lessons to be able to allow the community to gain skills that looking forward, they can lead to job opportunities. We want to empower the next generation to be able to act as a stewards of their ocean. We want as well to be able to the community to understand the link between the coral, the ocean, and the need to protect it. And in terms of the impact of BEAM globally, many coral restoration methods, they are expensive. So BEAM wants to demonstrate that effective restoration techniques can be low cost, using affordable techniques that community can implement, can be scalable, that use simple method that can be replicated with the available techniques, that are adaptable globally, that are flexible approaches that are suitable for different reefs and different community contexts, and that are natural based, because we are not using a traditional coral restoration method. We are allowing the native coral to grow. So basically, we are working with nature to support resilience and to be able to create a healthy reef. And we strongly believe that the restoration solution must work also where resources are limited. And in terms of next step for us, we want to finish the plotting, setting up the restoration plots. After that, we are going to start monitoring and using ImageJ to understand what's happening. We would like to expand because, as I told, we are only working now with one of the dive sites. So we want to expand and work in other places and be able as well, for example, to compare this method in different depths to understand if the depth can increase or reduce the coral recruitment. We want to be able to publish a paper with the outcome of this data. And we want as well to strengthen a partnership with the scientists from Australia, other countries, as well as Mozambique. Our main goal is to restore these, in this case, the coral reefs that are crucial ecosystem for the Jangamo community, as well as inspire them to take action to their ocean. And thank you.
Thanks so much, Yudmila. It's so amazing to hear about all of your inspirational work and love the oceans. So another quick reminder to please add your questions for Yudmila in the chat, and we'll save them for the Q&A portion. Our final presentation is courtesy of Wave Moretto. She is our first place winner of the 2026 Ocean Innovator Award. So as Wave gets her slides pulled up, I will introduce her. Wave is a marine ecologist and PhD student at Oregon State University and an avid scientific scuba diver. She grew up just an hour away from California's beautiful Central Coast, quickly developing a love for kelp forests. Wave began her marine science career while earning her BS in marine biology at UC Santa Cruz. Afterwards, Wave continued to pursue marine science research at Scripps Institute of Oceanography, where she obtained her MS in marine biology and studied the effects of temperature on the diet and behavior of brown box crab, which is an experimental fishery in Southern California. Now, in her third year of her PhD program, Wave is part of Dr. Scott Heppel's lab, where she studies the role of kelp forests in sustaining Oregon's nearshore fish communities and has developed an innovative combination of scuba survey methods to document the seasonal changes in Oregon's kelp forest fish communities. Wave is also using environmental DNA to monitor fish community composition and working with the Hatfield Marine Science Center's Innovation Lab to develop an autonomous eDNA sampler for deployment in Oregon's kelp forests. WAVE works alongside the Oregon Marine Reserves team and Oregon Kelp Alliance to develop monitoring frameworks to ensure that conservation and management groups can continue to effectively monitor kelp forests in Oregon. WAVE is also deeply passionate about involving early career students in her research and communicating the findings of her research to the public. As such, she is working alongside teachers in the Oregon Marine Scientists and Educator Alliance to develop a curriculum on kelp forest ecology. WAVE intends to continue to pursue research that informs marine conservation efforts and plans to one day become a professor overseeing her own kelp forest ecology lab. I'm now thrilled to hand it on over to WAVE. Please take it away.
Awesome. Thank you, Katie. Thank you so much to Octo and Seashore Solutions for having me. It's an honor to be here. My name is Wave, and I'm a PhD student at Oregon State University in Dr. Scott Happel's lab, and I'm really excited to talk about my ongoing research, fish kelp and habitat health, redefining how we monitor Oregon's kelp forests. And so just a little bit about me and how I kind of got into kelp forest ecology. I grew up near the central coast of California, and when I was able to start scuba diving and really explore these ecosystems, that's how I fell in love. Seeing them from a totally new perspective underwater really heightened my appreciation for kelp forests, and I knew that I wanted to pursue research that would work on conservation and protection of these really vital ecosystems. And so I've luckily had the opportunity to do that through my PhD at Oregon State University, where I'm researching Oregon's kelp forest fish communities. In Oregon, our kelp forests are dominated by bull kelp, Nereocystis leuciana, which is a canopy-forming kelp species, meaning that it grows from the seafloor all the way to the surface. And very notably, bull kelp is an annual species, so it naturally dies off during the winter and then starts to grow again during the late spring to early summer. Unfortunately, Oregon's kelp forests and really kelp forests all along the US West Coast have been subject to many stressors in recent years. So notably from 2014 to 2016, we experienced a large marine heat wave known as the warm blob off of our coastline, where waters were warm for this really prolonged period of time. And this led to lots of shifts in marine species distribution and die-offs of many species. And we had another intense marine heat wave in 2019. And this was particularly impactful to species like bull kelp, which experienced stunted blade growth at temperatures exceeding about 14 degrees Celsius. And we also see decreased reproductive output in bull kelp at warm temperatures as well. And so this is one stressor that was really hitting kelp forests hard. And another one that kelp forests were experiencing was specifically impacting trophic interactions or the food web structure in Oregon's kelp forests. So we've got our bull kelp and other algae species at the base of the food web, and kelp is eaten by many different marine organisms, including purple sea urchins, which are known to passively graze in a healthy kelp forest ecosystem. And purple urchin populations are kept in check by predatory sunflower sea stars or Pycnopodia. So these large sea stars consume urchins and prevent them from overgrazing kelp. But what would happen if these sea stars were to disappear from these ecosystems? Well, unfortunately, that's exactly what happened in 2013 with the onset of sea star wasting syndrome, which wiped out sunflower sea stars and other sea star species all along the US West Coast. And with the loss of this predator, this caused purple urchin populations to skyrocket, and this increased grazing pressure on kelp forests. And so this combination of grazing pressure and thermal stress really decimated our kelp forest ecosystems, and we saw this massive shift from these once really densely forested ecosystems to something known as urchin barrens, where purple urchins carpet the rocky reef seafloor. And this loss of kelp forests was quantified in Oregon by a group called the Oregon Kelp Alliance. In 2024, they released a status report, and they present this really alarming statistic that from 2010 to 2022, Oregon's peak annual kelp canopy has declined by about 70%. And so we know that we've lost a lot of this vital kelp forest habitat, and what I'm really focused on now is what does this mean for fish communities that also reside in Oregon's kelp forests. And so we know that kelp forests function as fish habitat. They're federally designated as essential fish habitat as they provide critical refuge and foraging area for many different marine fish species, including rockfish, lingcod, cabezon, and even salmon. And in our neighboring states, California and Washington, kelp forests have been shown to support more abundant and diverse fish communities relative to those urchin barren systems. And in Oregon, many of our key fishery species are also linked to kelp forests. For instance, our recreational groundfish fishery is valued at just over $20 million annually, and black rockfish make up the majority of catches for that fishery, and black rockfish are our most common kelp forest fish species that we see. And despite this link, we have very minimal data on kelp forest fish surveys on the Oregon coast. And so why do we lack that kind of data? Well, I wanted to show this video, which was taken from sampling last year at one of our kelp forest sites. This represents a really nice day for doing field work on the Oregon coast. We have good visibility, about 20 feet, minimal surge and current, and lots of rockfish to count. However, when we returned to this exact same location just a few weeks later, it looked like this. We had a big phytoplankton bloom that reduced visibility to about 3 to 5 feet. And it's hard to tell from these photos, but there was also a lot more surge. And so when we have these intense blooms, it's really difficult to count fish that are moving around. And these phytoplankton blooms occur pretty frequently from about April through September off of the Oregon coast. And so that can really make doing fish surveys quite challenging. Another challenge that we face doing fish surveys or really any kind of scuba diving work on the Oregon coast is due to the frequency of intense storms that occur during the winter. And really, we get these big systems moving through our coastline all year round. Even just last week, we had this 15-foot swell period. And so Oregon is a really flat, exposed coastline that lacks any kind of bays or inlets like we have down in California or up in Washington and British Columbia. And so when we have these systems moving through, we really can't get boats out and do any sort of field work in the near shore. And so that combination of frequent low visibility in the summer and intense storms in the winter can make doing these scuba surveys really challenging in Oregon. I did briefly want to highlight that there are current nearshore scuba survey efforts. The Oregon Coast Aquarium supports the subtidal monitoring for the Oregon Marine Reserves and other projects from the Oregon Department of Fish and Wildlife. And the Oregon Kelp Alliance works with ReefCheck to monitor kelp forests and urchin barren systems as well. However, all of these surveys are fairly limited in terms of temporal coverage. They're typically a few days out of the year for the marine reserves and limited to the summer or early fall. And they target the marine reserves, which aren't necessarily kelp forest systems, and a lot of the work that Orca and ReefCheck does is more concentrated on the southern Oregon coast. In addition, these surveys tend to be fairly invertebrate and algae focused, as these are much easier to count as they don't move relative to fish. But we're working alongside these groups. I've gotten to go out and dive with all of them to identify and address knowledge gaps so that we can better figure out how to monitor fish communities in Oregon's kelp forests. And so that brings me to our overarching goals: optimizing methods to study Oregon's nearshore kelp forests and fish communities, and documenting the status of those fish communities so that we can establish a baseline in the face of future change. And so just to show you all where we're located, so OSU, or Oregon State University, is in Corvallis, Oregon, and we're monitoring at several sites along the Oregon coast. And so we're diving at Cape Foulweather, Cape Arago, and Port Orford, which spans a sort of northern to southern range of Oregon's kelp forests and varying levels of kelp forest spatial extent. And so our approach involves monitoring for at least 3 years. We began this project in the summer of 2024, and we are going out year-round sampling quarterly. So this includes sampling during the winter months, which is sort of that off-season for bull kelp, so that we can get an idea of fish communities both when kelp is at its peak in the summer and also when it's not very present in the winter. And we're conducting scuba surveys within kelp forests and adjacent urchin barren sites at each location. These surveys are 50-meter long transects, and we use a method known as stereo video, which is what you see that diver holding there. And then I follow next to them doing an underwater visual count, or UVC, which is a traditional method of tallying up fish on a data slate. So we record information on fish abundance, canopy and understory or subsurface kelp counts, invertebrate counts for key species like sunflower stars and urchins. We measure substrate type and relief height, as well as temperature and underwater visibility. And just to highlight stereo video, this method is fairly popular in tropical ecosystems, but is a little less common in temperate environments like kelp forests. But What we do is we film a fish with both left and right side cameras that are synchronized, and then we upload those paired videos into a program called Event Measure, and that allows us to get total length measurements for fish without ever having to catch them or handle them in any way. So it's a really great non-invasive method for monitoring fish communities. And so just to highlight a few of our findings so far, we've detected more fish using this stereo video technique relative to the underwater visual count. We found that kelp canopy cover, habitat structure, and water temperature all significantly influence kelp forest fish diversity. We see significantly less rockfish on reefs when kelp forests decline during the winter. And we see significantly more and significantly larger rockfish in our northernmost kelp forest site relative to our urchin barren locations. So we have been able to collect some of that first line data, particularly comparing kelp forests in the peak season relative to the winter. That being said, there are still a lot of limitations with this work, and one of the biggest ones is the overall ability to detect fish diversity. It's fairly low using both the UVC and the stereo video methods, as we tend to encounter a lot of large schools of black or blue rockfish, and we're not seeing a lot of other more cryptic species. And so one solution to this is using environmental DNA or eDNA. So this is DNA that fish or any organism sheds into the water in the form of scales, gametes, skin, waste, things like that. And so we're able to collect a water sample, extract the DNA from that, and determine whether a specific species was present or not in that sample. And it gives us another way to measure species composition that doesn't rely on being able to physically see a fish. And so what we've been doing is collecting water at depth and at the surface, filtering it, preserving and freezing it, and then extracting and sequencing that DNA. And so this can provide us another way to look at community composition. But this still doesn't really address the issue of being limited about days where we can get in the water. Our diving and boat days are still restricted by time, cost, and that effort. And we're also selective in our sampling to having good enough weather and sea state to be able to get a boat out and get divers in the water. And so ultimately, these discrete samples that we're collecting, although really valuable, are still only providing a snapshot. And so it would be really valuable if we had a way to continuously monitor the nearshore environment for fish communities. And so that has brought about us working to develop an autonomous eDNA sampler. So this prototype was developed by a former Hatfield Marine Science Center student, which is our marine campus, Genevieve. So this prototype is called Edulus, the Environmental DNA Underwater Long-Term In Situ Sampler. And so when she first developed this, it's a 4-channel system. So you can see in that photo, these 4 different samples will draw water in, pump them through a filter, and then that filter is preserved and then can be later collected. And so this is super valuable, but it still only allows us to collect 4 samples at a time. And so we're working with Hatfield's Innovation Lab to develop a sampler that can be deployed for about 1 to 2 months in Oregon's nearshore and collect samples daily or every other day so that we can leave this out basically in the time that it takes between us doing one dive and then getting out again. And so it can kind of fill that gap when we don't have decent weather conditions to get out and do scuba surveys. We're also designing this to be modular so that a diver can pop down, swap or take out those full filters and then swap them in for blank ones. And so that way it can be easily serviced by divers. And I did also want to highlight that there are commercial eDNA samplers already on the market, but they cost anywhere from about $10,000 to $50,000. And we're working to get this down to a raw parts cost of about $500 to $1,000 per sampler so that it's much more available to different NGO and citizen science user groups. And so once this is complete, the design and parts list will all be made open source, and we want to make this also very user-friendly so that the group that wants to use it can simply set the kind of sampling frequency and amount of samples collected and let it run from there. And so to wrap up, I wanted to highlight this new nearshore monitoring framework that we've developed for Oregon relies on kind of these 3 crucial parts. First, because of that annual cycle of bull kelp, sampling year-round so that we can look at how fish communities change seasonally when kelp is at its peak and when there's not much kelp available. using tools that have been used more commonly in other systems like stereo video to non-invasively measure fish abundance as well as size. And then lastly, this implementation of the autonomous eDNA sampler so that we can fill those gaps in between our ability to get out and do in situ scuba dive surveys. With that, I'd like to thank everyone who's made this project possible so far. A huge shout out to scientific divers Alexander Bayless and Olivia Boyson, because I could not do any of this work without them. My advisor, Dr. Scott Appel, and the rest of the Appel lab. And I also want to really give a big shout out to Sea and Shore Solutions for the funding that they provided will help go directly to further development of the Edulus autonomous eDNA sampler. And if you have questions or want to contact me, this is my info, and you can also check out our lab Instagram. Thank you.
Amazing presentation, Wave, and all of our winners. We're so excited to have you all here. So it's now time to move over onto some questions and answer time. So Andrea, the first question I'm going to give to you, it's from one of our participants. How many pearls do you need per net? They're not quite sure about the average size, so if you could say that again. And how much do they cost to produce?
Hi. Yeah, that's a very good question. We have a minimum distance that is between 35 and 40 centimeters. That is the minimum distance that the animal can understand that they cannot pass through it. So the number of pearls will depend on the net size, but we are always following this distance. So yeah, and the cost, it depends also of the size of the net. In Germany, it's around 2-3 times a normal net, so it's more expensive. But we hope that with this new design pearl, the price will reduce the time, so consequently the price as well.
Great, thank you. Yudmila, the next question's for you. Have you had any issues with algal fragments resettling onto the reef? The participant noted that that's a problem that they see in the Caribbean.
For us, it's not really a problem. We just have, because we don't have the normal fish that will feed on algae, we just have this huge grow on algae. So, but it's not our case.
Great. Thanks, Yudmila. All right, Wave, question for you. Do urchin feeding fish move into the urchin barrens, or is there just a general lowering of biodiversity in these areas?
That's a great question. So in Oregon, we don't have many urchin feeding fish. We do have wolf eels, which will tend to eat urchins, but that's not something that at least we've seen a lot with our surveys. I know down in California, for instance, you'll have like California sheephead, and those are known to eat urchins. But I think kind of some of the issues is that the urchins in those barrens tend to be, if they haven't had anything to eat for a while, they go into this sort of dormant state, and so they can like subsist with really minimal prey, and so they get, fairly small. They don't have a lot of like food content, and so this is an issue at least with like sea otters eating them. They kind of figure out that they're not super nutritious, and they won't eat those barren urchins because they're not a good source of food.
Great, thank you, Wave. Andrea, back to you. Do you have any plans to monitor catch rates of fish to see if they decline with the pearls, or they're thinking about ways that fishermen may push back on the adoption of this technology?
Yes, so we first start with it. So we did a trial in Germany regarding flatfish. So we have a paper on it. I didn't put it in the slide, but yeah, it's free access. And it's not affecting at all. It was the same result in Denmark with cod, and also in Iceland with cod was the same. It's not affecting.
Thanks, Andrea. Yudmila, why did you choose this method instead of traditional coral transplantation methods?
Oh, that's a great question. First, because in our area, we needed to understand what is happening, and from a lot of our data monitoring, we are starting to see a lot of disease. So starting to doing transplantation in a place that is diseased is a little bit difficult because we are going to increase the amount of disease, and we don't want that. And second, as well, because it's expensive, and in the area that I work, that I'm based off, is quite rural, and the resources are limited. So it was not an affordable technique and was not an option for us. So that's why we needed to go for this innovative technique. Thank you.
Thanks, Yudmila. Wave. Regarding the eDNA, are you using amplicon sequencing? Is this just to measure fish diversity, or are you also trying to quantify or correlate fish abundance?
Yeah, so another great question. So we are focused on just doing presence absence right now, so more focused on diversity and not abundance. So we're doing metabarcoding. We're using there's a specific primer that's been used for or developed for rockfish called Mysebastes. And so since Oregon's kelp forests are really dominated by those species, we're using that primer. We're also using the MyFish, which is like the 12S primer as well. But we're definitely open. There's lots of groups in Oregon that are interested in species like abalone and sunflower stars and things like that. So we kind of just have the funding to focus on fish, but certainly open to looking at other species too down the road.
Great. Thank you, Wave. So I'm going to do one more question for each of our winners here. So Andrea, back to you. Do you think that PearlNet could also function in a river environment to prevent Bodo dolphins from entanglement in the Amazon?
Yes, we did our simulations comparing freshwater and seawater, and we didn't find any difference. We didn't test it yet, like on the field, but based on the simulations, yes, definitely.
Amazing. Good to know, Andrea. Yudmila, why did you select the specific coral species for the study? Was there a specific reason for choosing those coral genera?
Yes, first because they are the common coral genera that we have in the area, and as well as because they are both species, they contribute a lot for the reef formation and the three-dimensional structure of the coral, and both are really good in resilience. They are both resilient, and they help a lot in the coral recovery. So that was the reason why we chose those two genera.
Thank you, Yudmila. Okay, Wave, last question here. Are you engaged with any other kelp projects along the Pacific coast?
Yeah, so mostly just those based in Oregon. So the Oregon Kelp Alliance is doing kelp restoration on the South Coast. So they're putting out these sort of seeded lines with little baby kelps to really help out in Port Orford, because that's where we've seen a lot of the majority of our kelp loss on the coast is in that region. And so something I'm working with another grad student in our lab, Olivia Boyson, to do is actually sample the juvenile rockfish communities that are using those artificial kelps or those outplanted kelps relative to natural ones. And so that's kind of one project that, or one other group that we're working closely with, but definitely open, especially I know there's a lot more groups down in California, up in Washington. And so always happy to collaborate and hear from other folks.
Great. Thank you so much, Wave. And to that note, I saw that all the winners posted their emails in the chat. So for everyone on the webinar, if you're interested in reaching out to them directly, please do so. Wave, I'm going to ask you one more question before wrapping up here. And that is, could you expand on how and where the autonomous eDNA sampler can be used beyond your PhD research?
Definitely. So, as I mentioned, this was initially developed or is initially being developed for use in Oregon's nearshore, but really this can be used in any kind of marine environment. Just as some examples of other places we're hoping to use this, our lab has ties to sampling in the Cayman Islands as well as in Palau, so in tropical like coral reef ecosystems, to look at those fish communities. But really, any citizen science NGO group group, the goal is to make this affordable enough that those folks could use them and also very user-friendly. And so, like I said, we'll be putting that parts list and design publicly accessible using GitHub when it's done. And so stay tuned for that.
Amazing. Thank you. All right. Well, fantastic, everyone. This brings us to the end of our Q&A portion. So please join me in thanking all three of our amazing winners for sharing their innovative efforts with us. We encourage anyone interested in learning more to reach out directly to Andrea, Yudmila, and Wave. The recording of this webinar will also be posted online for future reference and sharing. Sea and Shore Solutions is looking forward to our next Ocean Innovator Award, with the winners announced in early 2027. So please don't forget to sign up for updates using the link in the chat so you don't miss it. So Deanna will post that link again. And with that, everybody, thank you so much for attending. This concludes our Ocean Innovator Awards webinar. Thanks for joining, and we look forward to the next round of innovators in 2027.